Good afternoon. My name is Modjadji, and I'll be actually taking you on a very different route from what everybody's been talking about. When everybody sees the big sharks and everything, I look at the big sharks, but then I get wowed at the little guys that hanging on to the sharks. So, and I'll be doing Siphonostomatoid diversity on mobulids of the KwaZulu-Natal coast. And this was part of my MSc, which I was able to finish, thank goodness. I'm graduating on the 24th of May, so hallelujah. Okay, we all know that in order to be able to conserve our marine biodiversity, it's very essential to understand the biology, the systematics, and the behavior of the marine biota. And there's very little knowledge, especially in South Africa, about the diversity and distribution of marine invertebrates, especially the copepods that are symbiotic with different hosts, ranging from invertebrates to marine mammals. And some of the most symbiotic copepods, we find them in the order Siphonostomatoid. There's actually 6 orders, so most of them we find in the order Siphonostomatoid. In this order, we have 39 known families of copepods, and in those 39 known families, we have found out that 22 occur in symbiosis with invertebrates, while 17 occur in symbiosis with vertebrates. And 11 out of those 17 have been reported from elasmobrowns. This is just This is just to show you all the different shapes and sizes of all the symbiotic copepods that we actually run into. Some of you might have actually seen them on the sharks that we've worked on. South Africa is very rich in marine biodiversity, and it has about 210 contributing species, and a small percentage, a very small percentage of those are mobulid rays. These rays are widely distributed in warm, temperate, and tropical areas across the world's oceans, so we find them almost everywhere. They are characterized by large pectoral fins, slender whip-like tail, a slender whip-like tail and elongated snout or rostral fin on each side of the head. That's the name devil rays. So they look like the devil in a way. There's actually 11 known species at the moment in the whole world, and only 7 have been reported from South African waters. And in those 7, we have technology. We have the 2 that belong to the genus Manta. We have the giant manta, Manta biorostris, and we have the reef manta, Manta alfredi. Those are the ones that have been reported from South Africa. And the remaining 5 fall under the genus Mobula, which we find the longhorn devil ray, Mobula eregoodootenkee, Mobula thurstoni, Mobula japonica, Mobula kuhlii, and Mobula terrapacana. Okay. What we know is the biology of mobulids it remains poorly known because of the difficulty to correctly identify the species in the field. Sometimes when you see them in all a bunch, it's very difficult to actually distinguish between the different species. So it's very difficult, and they're very uncommon. They're rarely seen and caught, and thus they're not very well studied. So if anybody's interested, they can actually just take up these devil rays to study. In South Africa, copepods that have been reported from mobulid rays include 4 species distributed in 4 families. So in one family we have, in the family Eudactylinidae, we have Eudactylina oliveri that's been reported. And in Caligidae we have Caligus elongatus. Both of these have been reported from the shorthorn devil ray, Mobula kuhlii. And then we have 2 more species that have been reported from Manta biorostris. So we have Antiferus laminipes here, and then we have Anthosoma grassum. Okay, usually it's very hard for us to actually convince people to understand what it is that we're trying to do, because where we come from, there's not even a shark nearby. So when we tell people that we're actually doing parasites of sharks, they don't actually get it. Why? I mean, what's the point? Why do you do them anyway? And then we actually get a very interesting question: do you eat them? So, well, I don't know if anybody has eaten them, but there have been reports of people making soup from them. So I don't know. I've never had any copepod soup, so I don't know. But the truth is, there's very little knowledge about the biodiversity of invertebrates and distribution and parasitic invertebrates in South Africa. That's a known fact. And when we look at copepods, compared with the 2,800 species reported worldwide, less than 10% have been reported from South Africa. And that's only usually due to the fact that there's only one qualified copepodologist working on symbiotic siphonostomatoids from elasmobranchs in South Africa. There's only one person working on that who's qualified. And right now I make half of the qualified. So at least now there's 1.5 qualified people working on it. So that's a little something. And in that way, marine symbiotic copepods represent an unexploited opportunity for researchers in marine biodiversity and especially copepodology in South Africa. So we do need more people if you're interested. Okay, the main aim of this study was to investigate the siphonostomatoids we find on mobulid rays, to examine those mobulid rays for the presence of those siphonostomatoids, and to identify each of those siphonostomatoids by comparing them with previously described species, by dissecting and drawing specimens where it's very difficult to actually identify them, so we do the drawings. And furthermore, we describe the dissected specimens and elaborate on ill-described features. The thing with copepods is most of them have been done like a long time ago. We have a genus of copepods where the most recent literature is 1967. So that's like, so there's a lot of elaborating on ill-described features because some of them are just, I mean, they've been done in the old days, so it's not very easy to understand them. Okay, we were able to collect 40 mobulid rays, of which 31 of them were Mobula kuhlii, 2 of them were Mobula eregoodootenkee, and 7 of them were Manta alfredi. Okay, so this is what we do. When we get the hosts, the different types of hosts, we immediately examine them on the outside to find which copepods have been attached on the outside. And then we cut out the gills and page through them, through the filaments, like we page through a book. So we just go each filament because they actually embed themselves in between the filaments. So we just page through those filaments. And after that, the copepods are fixed and preserved in 70% ethanol. And we view them under the microscope to try and identify them. And if we cannot identify them, that's where we make all the drawings and try to see if we can identify them better. Okay, this is what we found. We found that more than 90% of all the examined hosts were infected with different types of symbiotic siphonostomatoids. And from that, we were able to identify 13 different species distributed over 5 families. In the first family, Caligidae, we were able to get 7 species. In Eudactylinidae, we were able to get 3 species, Croeridae, Diabolophila, and Pandaridae. We were only able to get a single species from each of those families. Okay, just to start with the family Caligidae. In this family, we found Caligus chrysophrisi. This species has only been reported from the yellow sea bream Rhabdosarcus sarva, which was caught off Kerala in India. So this marks the first record of this species on an elasmobranch. And we were able to collect this from Mobula kuhlii and Manta alfredi. So this is the first record of this species on an elasmobranch. And then the second genus that we were able to identify is Pupillina. Now this is the 3 species that are known within this genus. So we know that there's Pupillina flores, Pupillina minor, Pupillina brevicoda. These were the species that are known, and we found that these species are actually specific on mobulid rays. So they've never been reported on any other species of shark or ray or whatever. And these two are quite distributed within the Mobula genus, while this one has only ever been reported from a Manta species. Okay. And these were the ones that we were able to collect. We collected 3 species, and we were able to identify and describe 2 new species, of which the paper was only published actually yesterday. So, so it's safe to actually talk about them now. Okay, one of them was Dippenaar kuhlii, and the other one was Dippenaar merera. This other one, we were able to see that it is a Dippenaar species, but we only had like 1 specimen from a host. So it's not really, we're not really convinced yet, and we can't really say it's a new species or whatever. So we're still trying to find more samples on these ones, and we collected them from Mobula kuhlii and Mobula eregoodootenkee. So if you ever find any parasites on Mobula kuhlii and the other one, that would be very much great. And then we were able to find 3 more species that we were also unable to identify because of the small sample size. And the interesting thing about these ones are they do look like Dippenaar species that we've identified, but we also found that they're actually very different. So if we find more samples, then we'll be able to actually conclude on what actually they are. And then in the next family, we were able to identify 1, 2, 3 species. This one has been reported from South Africa before, and these 2, Eudactylina, diabolophila, and Nemesis, this is the first time that they're actually being reported from South Africa. And this one is Nemesis. Nemesis has only been reported from Daciatus, the diamond rays, Daciatus. Yeah, well, yeah, the string rays, yeah, those ones. Yeah, so this is the only species that has been reported from other rays as well, but first time ever on a mobulid ray. And then in the family Groyeridae, we were able to identify Groyerina mobile. Unfortunately, I couldn't find a picture of this one. It's a very small little copepod, so it's very hard to get a picture of it. They are also specific to mobulid rays, and what's interesting about them is that they don't, the other ones infect the gills and the body surface and everything. These ones infect the nasal lamellae, just to show you where they just embed themselves in there. They're very small. And we found them infecting Mobula kuhlii. Okay, the last 2 families, we have the family Bandaridae, where we get Antiferous laminipes, and we have the family Dippenaar, where we have Anthosoma grassum. Anthosoma grassum is not really restricted to any particular shark species. We find them everywhere. So we collected them from great white sharks, we collected from all the other sharks as well as mobulid rays. While this one has only been reported from mobulid rays. So all of them are quite specific except for Anthosoma grassum. Okay, back in the day, mobulid rays were not carefully studied for copepod infection, and the copepods that were reported from them were probably found by accident. So they were not very well studied back in the day. And imagine now we have been able to identify at least 13 species distributed over 5 families from only looking at 3 of the 7 that occur in South Africa. Now imagine if we were able to look at all of them, how the number would just go up. Like I said, this is the first record of 10 of the species that we were able to identify. So 10 out of 13 were first records ever from South Africa, and some of them were first records from the entire world, actually, in a way. South Africa, like I said, it's very rich in aquatic species, with about 210 chondrichthyan species. All of them are probably infected with a large diversity of copepods, and I'm pretty sure that almost all of you who have worked on sharks have seen all these copepods before. So almost all of them have the ability to be infected by rays. The only shark species that we've worked on and we didn't actually find anything over the past 3 years is the angel shark. So that's the only one that we've worked on and we haven't found anything. So by studying the distribution and diversity of symbiotic copepods and mobulids in South African waters, we make a small but significant contribution towards the knowledge of symbiotic copepods in general and the biodiversity of marine invertebrates in South African waters. Okay, just to thank a few of the people. I don't think this project would have taken off if it wasn't for KwaZulu-Natal Sharks Board. I mean, that's exactly where we get our samples and everything. Marine megafauna, Dr. Andrea Marshall, I mean, she helped us with, the identification of the manta race, because usually we always thought that manta virosis was the only one that occurred in the Indian Ocean. So she was helpful in that way. And also marine dynamics, also oceans research. I mean, they invited us here for a whole month to actually get samples from them, so which is also good. Thank you very much. And thank you.